Hello children. After discussing about the characters of living organisms and taxonomy in yesterday's chapter called Living World, today we'll be in the job of classifying all these living organisms based on some criteria. Yes, you guessed correctly. We are discussing a chapter which is behind me, that is biological classification. We are going to divide this chapter into 4 to 5 units. First is introduction, then for few minutes we'll be discussing about Monera, Protista, fungi, and plants and animals along with viruses. So without wasting any time, because it's a big chapter, so we will go a little faster and try to complete this in a record time. Right. Now, very important is the first is the types of classification. Broadly, first listen to me, then we'll be seeing what is there on the board. Two types of classifications. One based on science, scientific, and another not based on science, that is not scientific, non-scientific, better way of saying. If we classify plants into useful, unuseful, and poisonous plants, then it is said to be non-scientific because there is no science involved. And if you classify living organisms based on certain rules or based on certain criteria, you call it as scientific. And we have got 4 to 5 examples of scientific classification, starting with the first one, Aristotle. Aristotle classified plants into herbs, shrubs, and trees based on nature of stem, so therefore it is scientific. He classified animals, those with red blood and those without red blood, so animals and plants. So Aristotle classification can be categorized as first scientific classification. Then Linnaeus classification, he classified living organisms into 2 kingdoms, kingdom Plantae and kingdom Animalia. Basically, taken criteria is cell wall. Then Whitaker, he classified living organisms into 5 kingdoms, which we'll be discussing in detail. All right, and these are the 5 kingdoms: Monera, Protista, fungi, Plantae, and Animalia. Calvo's classification is a 3-domain classification. So a taxonomic category which is higher than kingdom is domain. So the 3 domains, and the 3-domain classification, archaea, bacteria, and eukarya. And 6 kingdom classification. He divided archaea from bacteria and gave 2 separate kingdoms, archaea, bacteria, and the remaining 4 is similar to Whitaker's classification. So we'll come back to the first classification in detail, children. We'll start with Linnaeus classification. 2 kingdom, we already discussed about that. The criteria is the cell wall, generally cell wall, gross morphological characters, but nature is cell wall. What are the merits? Definitely the scientific classification, and is it easy to understand, is also said to be taken as a merit. But the demerits are more in number. Large number of organisms did not fall into any category. Euglena, Linnaeus did not keep in any kingdom because it has got both plant and animal characters. Slime moulds, he did not keep in any kingdom. So therefore, majority of organisms did not fall in any kingdom is one of the demerit. Prokaryotes are not distinguished with eukaryotes. Plants are kept along with bacteria simply because both of them have cell wall, but bacteria is a prokaryote, plant is a eukaryote. Unicellular organisms are kept along with multicellular. Chlorella, chlamydomonas, diatoms, dinoflagellates, they're all kept along with plants which are generally multicellular. So therefore, this is also a demerit in Linnaeus classification. Photosynthetic organisms are kept along with non-photosynthetic, hard-working plants, and parasitic bacteria. Both of them are kept together just because both of them have cell wall. That is little unfair. So therefore, these are the 4 I call it as limitations in Linnaeus classification. Quickly, if you run through, children, in Linnaeus classification, in which kingdom they are kept: bacteria, blue-green alga, diatoms, dinoflagellates, all are kept under plants because they contain cell wall. Euglenoids and slime moulds did not fall in any category. Protozoans, they do not have cell wall, therefore animals. Fungi, they contain chitin, very important. They are plants. Yeast contain chitin, plants. Chlorella, chlamydomonas, spirogyra, they all have cell wall, plants. Spirulina is a blue-green alga. So Blue-green algae also have a cell wall, but there are some organisms Linnaeus did not classify in either of them. So bacteria are kept under plants as they possess peptidoglycan, which possess two types of amino sugars, namely NAG and NAM. Fungi and yeast are kept under plants as they possess chitin. Diatoms, dinoflagellates, chlorella and chlamydomonas are kept under plants. Protozoans are kept under animals as they lack cell wall. Euglenoids, slime molds are not kept in any kingdom. So that's about Linnaeus classification. In fact, we gave a nice description with regard to Linnaeus classification, nice facelift with regard to it. Without wasting any time, we'll go to Whittaker's classification. Please remember the year 1969. The type of classification is phylogenetic, and there are 5 kingdoms. Look here, Whittaker is a very smart man. I'll tell you why. Because the demerits of Linnaeus classification are taken as a criteria by Whittaker to construct new kingdoms. What was the demerit of Linnaeus classification? Prokaryotes are kept along with eukaryotes. Whittaker separated prokaryotes and gave a kingdom Monera. What was the second demerit of Linnaeus classification? Unicellular organisms are kept along with multicellular. Whittaker separated all unicellular eukaryotes and gave a separate kingdom called Protista. What was the third demerit in Linnaeus classification? Autotrophs and heterotrophs are kept in same kingdom. Whittaker separated heterotrophic fungi and gave a separate kingdom. So therefore, it's very easy to understand Whittaker's classification. So Monera, Protista, fungi, plantia and animalia are the 5 kingdoms in Whittaker's classification. And lichens are not kept in any kingdom. Viruses, prions and viroids are acellular creatures which are not kept in any kingdom. Cellular means lichens, acellular means this. Now let us put the same table, this time Whittaker's classification. Bacteria, blue-green alga, mycoplasma, spirulina, all of them are prokaryotes, therefore monerans. Look here, diatoms, desmids, dinoflagellates, euglenoids, slime molds, protozoans, chlorella and chlamydomonas. They all are unicellular eukaryotes, hence they are protistants. Watch out for these two, chlorella and chlamydomonas. Time and again you made mistakes, children. So these two are actually protistants according to Whittaker's classification, simply because they are unicellular and eukaryotic. Now, fungi, yeast, they are kept under kingdom fungi. Alga to angiosperms plants. Spirogyra, don't get confused between spirogyra and spirulina. Spirogyra is a plant, whereas spirulina is a moneran. Okay, no confusion. Porifera to mammals, they're animals. I think these are very simple. Bacteria, blue-green algae and spirulina are kept under Monera at the prokaryotes. These are all very important. These two I highlight again. Chlorella and chlamydomonas are kept under Protista at the unicellular eukaryotes. This is a table which looks like a repeated table because I kept both Linnaeus and Whittaker side by side. If you want to see for a minute, we'll do it. Bacteria, plants in in Linnaeus and in Monera. Diatoms, plants, Protista. Dinoflagellates, plants, Protista. Chlorella and chlamydomonas, plants, Protista. Euglenoids and slime molds did not find any kingdom. So here Protista. Protozoans, animals, Protista. Spirulina, Monera. Spirogyra, plants and plants. That's very easy one. Chalo children, now we are in the last 2 minutes of concluding the introduction part. What are the 5 criteria picked by Whittaker? Cell structure, body organization, nutrition, reproduction, and also phylogeny. The first important one we're going to talk about is cell structure. First of all, what is the meaning of cell structure? Whether it is prokaryotic or eukaryotic. What is the meaning of prokaryotic? You know that. Primitive nucleus, no nuclear membrane, no nucleolus, no histones. Primitive nucleus, circular DNA. That is the character of prokaryotes. Very important. Monerans are prokaryotes, and the remaining all, Protista, fungi, plants and animals are eukaryotes. The same thing I have written here. Monerans are prokaryotes, Protista, fungi, plants and animals are eukaryotes. Then comes body organization. You're going to ask me, what is body organization? Means cells, tissues, organs, organ systems. This is the organization, isn't it? So cells are seen in all 5 kingdoms. Tissues started from fungi, loose tissue organization. Organs started from kingdom Plantae, and organ systems, digestive system, respiratory system, they are seen in kingdom Animalia. So that's the same thing I've written here. Monera only cells, Protista only cells, fungi cells and loose tissue, plant cells, tissues and organs, animal cells, tissues, organs and organ systems. Now the most important criteria picked by Whittaker for classification is nutrition. And this is a little tricky part of this entire Whittaker's classification. Should be careful what type of nutrition is seen in each kingdom. Monera. Monera shows both autotrophic and heterotrophic. When we go to Monera, we'll be discussing autotrophs are again 2 types, photoautotrophs and chemoautotrophs. That is characteristic of only monerans. I mean chemoautotrophs. In no other kingdom, you're going to see chemoautotrophs. And heterotrophs, they may be saprophytic, parasitic or symbiotic. So you see very much huge nutritional diversity. Very popular question in NEET with regard to monerans. Coming to Protista, there may be autotrophs and heterotrophs. Diatoms, dinoflagellates and euglenoids are autotrophs. Slime molds and protozoans are heterotrophs. Coming to fungi, it is a kingdom which is exclusively heterotrophic, just like animals. Heterotrophs only. They may be saprophytic or parasitic. Slime molds in your textbook clearly given. It's a saprophytic, feeding on dead and decayed organic matter. They may be parasitic also. Albigo, which cause white rust, is a very popular question asked. All right, that is a parasite. Coming to plants, they're autotrophic. Coming to animals, they're heterotrophic. They may be even saprophytic, according to the NCERT textbook. In the table, saprophytic is also a word. Holozoic, ingestive mode of nutrition, is also distinct of animals. Now, Whittaker's classification has given more importance to nutrition. Bacteria has 2 types of autotrophic nutrition. We'll come back to this point after a minute. And bacteria shows 2 types of photoautotrophs also, anoxygenic and oxygenic. Just wait for a minute, we'll be discussing more on that. Then coming to the next criteria, children, reproduction. If you look at unicellular organisms like Monera and Protista, the type of reproduction which is very common is asexual. In lower organisms, asexual reproduction is very common. But when you look at higher organisms like kingdom plants and kingdom animals, sexual reproduction looks more prominent in comparison to asexual reproduction. Fungi, asexual reproduction and sexual reproduction both are seen, but asexual spores are mostly formed even in fungi, just like Monera and Protista, which is very important. Now coming to the most important part, children, is about the cell wall. Now be careful, because at 2 or 3 positions will come across cell wall. First, Monera. Cell wall is peptidoglycan with amino sugars NAG and NAM. They alternate with each other. To one side of NAM, N-acetylmuramic acid, you see the presence of a polypeptide chain. So it means not only you see 2 types of amino sugars, NAG and NAM, you also see a group of amino acids attached to NAM. So you see both amino sugars and amino acids in cell wall is a very good question which can come with regard to the cell wall composition of monerans. Coming to what is called Protista. They have a cellulosic wall, and mind you, in diatoms, silicon is present in cell wall along with cellulose is a very popular question. Coming to fungi, even in biomolecules, this point is there. Exoskeleton of arthropods and fungi contain chitin. Chitin, just like peptidoglycan, contain an amino sugar called NAG, N-acetylglucosamine. But it doesn't have NAM and it doesn't have amino acids, is an important point to note. Then plants contain cellulosic wall and animals doesn't have any wall. Well, what did I do, children? Just like look here, I have no time to put some MCQs. Therefore, what I did is I kept some lines like fill in the blanks where you can just run through and see how much good you are in remembering it. Just please go through this. Primitive nucleus monerans, true nucleus in all, nucleolus except monerans all, nucleoid monerans, cells are seen in all, tissues are seen in all except Monera and Protista, loose tissue fungi, organs, plants and animals, organ systems, animals. Cell wall is seen in Monera, Protista, fungi and plants, no animals. All right, amino sugars in cell wall, Monera and fungi. NAM, amino sugar and amino acids in cell wall, Moneran. NAG, amino sugar in cell wall, Monerans and Protistans. 70S ribosomes, all 5 kingdoms. Flagella without 9+2 arrangement of microtubules, very popular question. All right, that is in Monerans. With 9+2 is seen in the remaining all. Autotrophic nutrition, Monera, Protista and plants. Oxygenic photosynthesis, Monera, Protista and plants. Anoxygenic is Monerans. We'll discuss that in a minute. Chemosynthetic nutrition is very unique of Monerans. Heterotrophic nutrition, Monera, Protista, fungi and animals. Saprophytic nutrition, same, Monera, Protista, fungi and animals. Parasitic nutrition, Monera, Protista, fungi. Even there are some animals which are parasite. Even though in NCERT book, the word parasite is not given for Animalia. Holozoic, ingestive mode of nutrition. They gave only for animals. There are some protistans which can also ingest. Holophytic, absorptive, fungi and plants. Absence of zygote. Zygote for the first time is seen in Protista. Remember that. So, Monerans. Presence of zygote. Respiration and copulating organs as in animals. Indeterminate growth is seen in plants. Intrinsic growth, all 5 kingdoms. Metabolism, all 5 kingdoms. Consciousness, all 5 kingdoms. That is the reason these 3 are said to be defining properties. I'm not saying growth is defining. Intrinsic growth is defining. That took a little time for us to get rid of the introduction part to biological classification. I'm pretty sure that you understood the areas which you have to highlight and try to remember. The notes which I've kept on the board will be very handy to go through it such that you can remember these things in the best possible manner along with the notes given by your teacher. All right, now Kingdom Monera. We'll start with Kingdom Monera, children. So we'll pick 10 minutes time here and get rid of this Kingdom Monera once for all. All right, it's a very easy unit. Try to follow what all I'm saying. Habitat. It is seen in soil, water and air. You can even call it as ubiquitous or omnipresent. And simple in structure, only single cells. Complex in behavior. Your textbook says complex in behavior. What do you mean of complex in behavior? There's a huge nutritional diversity. I can exemplify this as a reason why this have complex in behavior. And there are different types of respiration, different types of nutrition, and that is the reason why you say complex in behavior. The sole members are bacteria. Is it a prokaryotic kingdom? Yes. It's a primitive nucleus is seen. That is the reason. Cell wall, we already discussed NAG and NAM. Membrane-bound organelles are, look here, cell organelles are absent, wrong. Ribosome is a cell organelle which is present. Membrane-bound organelles absent, correct. Ribosomes are of 70S. Chromatophores, very popular question. It is seen in blue-green alga. All right. They're extensions of membrane. Nuclear membrane and nucleolus are absent. Nucleoid, this is an important point. So look here, any line which is present in your NCERT textbook in more than one chapter can be a potentially important question. Remember that. So nucleoid, it is a circular double-stranded DNA in large loops associated with non-histones and replicate at 2000 base pairs per second during binary fission. You know that. Plasmids, small circular double-stranded DNA. They regulate phenotypic characters used as vector in genetic engineering. Flagella lack 9+2 arrangement. Chlorophyll A and phycocyanin is seen. Histones are absent. Chain initiating amino acid is N-formyl methionine. Introns and exons are absent. And green sulfur bacteria possess chlorophyll. I'll talk about this. All right, chemosynthetic nutrition, iron and ammonifying bacteria. Anoxygenic photosynthesis, green sulfur. Oxygenic photosynthesis is blue-green alga. Water blooms, heterocyst is seen in blue-green alga. And primitive type of DNA transfer is a type of sexual reproduction which will come across. Coccus is a spherical bacteria which divide in all planes. Bacillus is a rod-shaped bacteria which contain endospores. Vibrio is a comma-shaped bacteria. Spirillum is a spiral shape of bacteria with tuft of flagella on both poles. Now, you got a little worried because I was looking a little faster. All right, now look here. I want to just go through this table first and come back to the lines which I have uttered a few minutes back. Look at this table, children. Prokaryotes are broadly classified into archaebacteria and eubacteria. Correct. Eubacteria are broadly divided into autotrophs and heterotrophs. Correct. Autotrophs are divided into photoautotrophs and chemoautotrophs. And heterotrophs are divided into saprophytes, parasites, and symbionts. Isn't it? First, let us get rid of this part. Saprophyte, which feed on dead. All right. In your textbook, lactobacillus is given. Bacillus is most common example of a saprophyte. Lactobacillus, curdling of milk, it's also a saprophyte. Parasite, feeding on living. In your textbook, there you canker in citrus. Xanthomonas citri is going to cause canker. Small swellings, pustules on the leaves. Canker in citrus and also on the fruits. Canker in citrus is caused by Xanthomonas citri. Then symbiotic, rhizobium bacteria. It's a rod-shaped bacteria present in the root nodules of legumes. That is the reason legume plants are very much rich in proteins thanks to rhizobium, which fix atmospheric nitrogen. So we got rid of heterotrophs. Easy to remember. Lactobacillus example here. Xanthomonas example here. Rhizobium example here. Now we go to autotrophs. Listen carefully. Photoautotrophs and chemoautotrophs. Let us get rid of chemoautotrophs. The name ample evidence. Autotroph means they take atmospheric carbon dioxide and they are going to combine with water to form glucose. And how it can happen in the presence of energy. So who's the source of energy? What are the source of energy? Oxidation of inorganic chemicals. Your textbook says inorganic chemicals get oxidized. Any oxidation process is energy releasing process. Energy is released. This energy helps in the synthesis of ATP. If it is an iron getting oxidized, iron bacteria, sulfur gets oxidized, sulfur bacteria, ammonia gets oxidized, ammonifying bacteria. They are all examples for chemoautotrophs. Question is, do they show photosynthesis? No. But why you say autotroph? Because they take atmospheric carbon dioxide and combine with water in the presence of energy liberated during oxidation of all these inorganic compounds to form glucose. That is the reason why you call them as autotrophs. Chemo, because inorganic chemicals get oxidized. Remember this. They help in recycling of minerals. So this is very important, chemoautotrophs. Coming to photoautotrophs. Broadly, I divide photoautotrophs into two types. Listen carefully. Those photoautotrophs which are oxygen liberating, example is blue-green alga during photosynthesis, means water gets oxidized, oxygen get liberated. Those photoautotrophs in which sulfur compounds get oxidized, therefore oxygen is not liberated, sulfur gets liberated. Even they are photoautotrophs, but we say that they show anoxygenic photosynthesis. Now I want to tell you, photoautotroph with oxygenic photosynthesis, blue-green alga. Photoautotrophs with anoxygenic photosynthesis is chlorobium, green sulfur bacteria, chromatium, purple-green sulfur bacteria. How can you forget Von Neal, who reported that oxygen is liberated only when water gets oxidized. And when sulfur compounds get oxidized during photosynthesis, there is no evolution of oxygen as we see in green sulfur bacteria. So therefore, easy to remember, photoautotrophs, oxygenic photosynthesis is blue-green alga, anoxygenic photosynthesis is chlorobium and chromatium. This table looks nice in your NCERT textbook. The same layout they have explained in the textbook, children. So therefore, it's more easier if you go through this and start reading the textbook lines. Now, coming to the first one, archaebacteria. This is a very, very popular question. Cell wall is made up of pseudomurine, hence survive in a very hostile conditions. This is one question which was asked maximum number of times in NEET. So therefore, cell membrane also contain branched chain lipids. Even they are also responsible in keeping the archaebacteria survive in hostile conditions or extreme conditions. But preference should be given to cell wall. In the absence of cell wall, you can say even plasma membrane is also responsible for that. Histones, proteins, interesting. They are present but simple. Normally histones are absent in prokaryotes, but archaebacteria contain histones. That is the reason why Carl Woese separated archaebacteria from remaining bacteria, because it has got some characters which are very unique, and one of them is histone presence. And the chain initiating amino acid is methionine, you know it. Common name, extremophiles. Why? Because they survive in extreme conditions. Methanogens, thermoacidophiles and halophiles are the three types of archaebacteria. Methane producing marshy areas, cow dung, and also in the gut of cow you're going to see methanogens. Thermoacidophiles, hot springs, and halophiles grow in salty areas. Respiration, I can't say always anaerobic, but mostly anaerobic. Methanogens, you have seen in a biogas plant 10 to 15 feet deep, and you're going to keep a floating cover, which is going to create an anaerobic conditions for methanogens to convert methane, to convert organic compounds into methane. You know that. Then coming to photoautotrophs, children. All right, archaebacteria is over. Now eubacteria. Within eubacteria, we discuss about photoautotrophs. The first one is cyanobacteria because they show oxygenic photosynthesis. Water blooms in polluted water. Very popular question. Unicellular, colonial or filamentous. Terrestrial and aquatic. Oxygenic photosynthesis, it shows. This point is important. Nitrogen fixing heterocysts. They're thick-walled cells, children. Why they're thick-walled? Because, look here, for nitrogen fixation, we require an enzyme nitrogenase. Nitrogenase is very much afraid of oxygen. It gets oxidized easily. All right, so therefore, nitrogenase should be properly shielded from high amount of oxygen. So, a thick-walled heterocyst is going to protect nitrogenase present in it from oxygen. Just like leg hemoglobin in legumes does the same job, protect nitrogenase by absorbing all the oxygen. So therefore, heterocysts, very important, nitrogen fixing cells which are thick walled. Why they are thick walled? Because they act as insulator for oxygen. Why like that? Because nitrogenase is very much sensitive to oxygen. What is nitrogenase? It's an enzyme which converts molecular nitrogen to ammonia. And is it used as biofertilizer in rice fields? Yes. In microbes chapter you studied. Anabina, nostoc, oscillatoria are the three blue-green algae which the farmers are going to add to the soil. They're going to plow. And these are going to do the job of converting molecular nitrogen into ammonia. So now the soil gets enriched with ammonia. Now if I transplant rice plantlets there, they develop properly, they grow normally, they grow efficiently because there is plenty of ammonia in the soil. Remember, blue-green algae with regard to rice fields are not symbiotic. They are not entering into rice plants. But the same blue-green algae in Cycas coralloid roots is symbiotic. So in Cycas coralloid roots, you see Anabina and nostoc. That is symbiotic. But with regard to rice fields, remember in microbes chapter we studied, they are not symbiotic, they are free-living. This point is important. See that you don't forget that. Chlorophyll A and chromatophores along with phycocyanin is seen here. Chromatophores, very important, blue-green alga. Nostoc, Anabina, Oscillatoria, Allocera are examples. Spirulina, it's an SCP. Spirulina is actually, now if you go to a pharmacy and ask for spirulina, you see a dark brown colored, grass green colored capsules they're going to give to you, and they're going to enrich you with plenty of protein. It's an example of a blue-green alga which is used as SCP. All right, single cell protein, spirulina. Don't get confused between spirulina and spirogyra. Spirogyra is a multicellular filamentous green alga, isogamy with non-motile gametes. That is spirogyra. This is spirulina. Trichodespium, outside chance of this question to come, red color of Red Sea. We already discussed this, children. Photoautotrophs and oxygenic photosynthesis. I told you, chlorobium and chromatium. Sulfur compounds get oxidized. That is the reason. Chemoautotrophs, we already discussed this. Oxidation of inorganic compounds and energy release is used to synthesize ATP. Heterotrophic bacteria, we already gone through it. Lactobacillus saprophytic, Xanthomonas, canker in citrus. Streptomyces is an antibiotic producer. Filamentous bacteria, children, not in NCERT book. You have got only penicillium in your NCERT book as an antibiotic producer, but even Streptomyces, filamentous bacteria, produce wide variety of antibiotics. Scientist's name is Waxman for the first time. Mycoplasma, pleuropneumonia-like organisms, witches' brooms in plants, pleuropneumonia in animals. These are the two diseases caused in plants and animals. They are facultative anaerobes. This point is important. It means that you and I cannot survive in the absence of oxygen. But what is very important about mycoplasma is, just like you and I, they are aerobes. But even in the absence of oxygen, they can survive, unlike us. So it means that they are facultative anaerobes. Sometimes anaerobes. Means mostly they are aerobes is an important point. Even E. coli, even yeast are examples of facultative anaerobes. They are smallest known cells. They are not sensitive to penicillin due to the absence of cell wall. because penicillin is going to prevent the wall formation during binary fission. When there are no walls, there is no effect of penicillin on mycoplasmal infections. That completes kingdom Monera. We took 10 minutes, no, little bit more time, but still. All right, we have completed in a record time, and we have discussed things in a manner in which it's more easier for you to remember. Chalo, now we go to the next kingdom, very, very important kingdom, Protista. It's a unicellular eukaryotic kingdom. It goes without saying. All right, so we pick 10 minutes time here to get rid of this very important kingdom called Protista. Diversified organisms, here you're going to see. Tissues, organs, and organ systems are absent. This point is very important. Boundaries are not well defined. Members exhibit overlapping characters with plants, animals, and fungi, making it difficult to clearly distinguish and classify them into a single group. And I think the Whitaker gave the name Protista because they're always protesting. That could be the reason. He did not disclose the reason, but I feel that could be the reason why he gave Protista, because they're very, very, what is called, in the sense, overlapping of characters are seen in them. Unique feature, diversity in form, even in nutrition. But when they say diversity in nutrition, one organisms which comes first will be monerans. Chemoautotrophs, photoautotrophs, within photoautotrophs, oxygenic, anoxygenic, saprophytes, parasites, symbionts. So when we talk about nutritional diversity, the one kingdom which comes to our mind is monerans. But even protistans, all right, can be the answer. In the absence of Monera, you can take Protista. Here also you see nutritional diversity. Plant protists, diatoms, desmids, and dinoflagellates, because they show plant characters. Plant animal protists, euglenoids. Plant animal fungal protists, slime molds. Animal protists, protozoans. Green protists means they perform photosynthesis. Diatoms, dinoflagellates, and euglenoids. Non-green protists, slime molds and protozoans. Cell wall is seen in diatoms and dinoflagellates. Cell wall is seen only in spore, slime molds. I think this table looks more easier after we complete Protista, isn't it? I think I was in a hurry to keep this table in front. But anyway, you have read all this, so therefore you can easily go through it and answer it. Cell wall is absent. Euglenoids, slime molds, and protozoans. Okay, in slime molds, only in spore cell wall is seen. Otherwise, in Plasmodium, there is no cell wall. Flagella is observed in dinoflagellates and euglenoids. 2 flagella in these. And protozoans, Trypanosoma and Leishmania, contain only 1 flagellum. 2 unequal flagella, dinoflagellates and euglenoids. Even in dinoflagellate, the transverse and longitudinal flagella are unequal. Single flagellum, Trypanosoma. Flagella is absent in diatoms, slime molds, and protozoans. Phytoplanktons, diatoms and dinoflagellate, they are the major producers of ocean. Chlorophyll A and C, diatoms and dinoflagellates. Mixotrophic nutrition, autotrophic and heterotrophic, euglenoids. Chlorophyll A and B, euglenoids. Multinucleate wall-less Plasmodium is seen in slime molds. We'll be discussing this after 3 minutes. And different locomotory organs, pseudopodia, flagella, cilia, that is seen in protozoans. What is seen for the first time in Protista? 9+2 arrangement of microtubules, gametes, zygote, isogamy, anisogamy, and oogamy. Okay? Chalo, we'll start with the first plant protist children, chrysophytes. Look here. What I did is I kept all this in one particular order such that you can remember it easily. First common name, then habitat, then cell wall, then chloroplast, then nucleus, nutrition, flagella, reproduction. Okay? For all the 5 groups of protistans, these are the common side headings. Okay, without wasting any time, we'll start with the common name. It's a plant protist, it's also a phytoplankton. Plankton means anything which are minute and floating are called plankton. Plankton means plant? No. Plankton is not plant. Or the meaning of plankton is not plant. Plankton means any minute thing floating on water. Phyto means plant nature. So there may be zooplankton also. So this is not a zooplankton. This is phytoplankton because this is a plant. Habitat, marine and freshwater. Important. Cell wall. Thick cell wall in the form of 2 overlapping shells like a soap box. Epitheca and hypotheca with silica. This point is important. Silica. Chloroplast, seen with chlorophyll A and C. Nucleus, true nucleus with membranes. Nutrition, photosynthetic, first major producers. Flagella generally absent, generally, okay? Now reproduction may be asexual or sexual. Oxospores are formed by the fusion of the two cells. Sometimes, you know, when binary fission occur, few cells keep what is called becoming smaller. All right, that description is not needed. Such two smaller cells may fuse to form oxospore. Special features: diatomaceous earth, passive movement on water, pollution indicators. They are the pioneer community for ecological succession operating in water. Both diatoms and dinoflagellates is the first community to colonize, all right, water. Diatoms serve as bioindicators, not much important. Navicular and pennularia, pinnate, cyclotella radiate, not important. All right, chalo, that completes what is called diatoms. We started with some side headings. Same side headings will be going for pyrophyta, that is dinoflagellates, common name, plant protist and phytoplankton. Same thing even for diatoms. Habitat, marine only, but for diatoms it is marine and freshwater. Cell wall, cellulosic plates, they are also in diatom cellulose but with silica. Chloroplast, A and C, they are also A and C, there means diatoms. Xanthophylls, this is important. Wide variety of xanthophylls are seen in dinoflagellates, which we don't come across in diatoms. Nutrition, photosynthetic, just like in diatoms. Second major producers, they're the first major producer. You can even call them as phytoplanktons, both diatoms and dinoflagellates. Mesokaryon, this word is important. Membrane is seen even during division, and chromosome remain condensed even during interphase and lack histones. This is in between prokaryotic nucleus and eukaryotic, hence we call mesokaryon. Flagella, two unequal, one transverse and one longitudinal. Both of them help in rotating and moving of this organism on water. Whirling whips is the reason why you call, because of this rotation. Reproduction, asexual and sexual. Special features, many types of xanthophylls. Red tide causing is goniolacs. Now what are red tide? All the goniolacs, you know, these dinoflagellates move in masses, all right, when they move in water in huge numbers. Two things they do: they clog the gills of fish in the process of their movement and cause a respiratory suffocation for them, and also they also secrete some neurotoxins like saxitoxin, which are harmful to the fish. That's an important point to remember. Noctile euca is a bioluminescent. Chalo children, now you found out the way to remember, so therefore we can decrease the amount of time in reading all this now. Euglenoids, common name, plant animal protist. What about diatoms and dinoflagellates? Plant protist. Habitat, standing fresh water. What about diatoms and dinoflagellates? Diatoms, freshwater and marine. Dinoflagellates, marine. Look here, red tide. You don't see red tide in the water body beside your house. Red tides are seen only in oceans. Remember, like that, only marine. All right, so therefore cell wall is absent. What about diatoms and dinoflagellates? It is present. Chlorophyll A and B. What about diatoms and dinoflagellates? A and C. Nutrition, photosynthetic and heterotrophic. What about there? Photosynthetic only. Heterotrophic also. That's the reason it's called mixotrophic. Nucleus, same. True nucleus in all three. Flagella, two unequal. One long and one small. Just like dinoflagellates, because in diatoms there are no flagella. Reproduction is asexual. Myonemes and pellicle, astaxanthin and stigma acting as photoreceptors. Two types of nutrition, two types of movement, reservoir, paraflagella body, outside chance for these statements to come in the exam. No need to remember them. Now we go to slime molds. This is little tricky. All right, I'll tell you why it is tricky. But before we talk about the cycle, the life cycle of slime molds, we put the same side headings here also. Common name, plant, animal, fungal protist. Habitat, it grows on dead and decayed organic matter, hence saprophyte. Cell wall, it is absent around plasmodium, but cellulosic walls are present around the spores. Chloroplasts are absent. Nutrition, saprophytic. Saprophytic nutrition. Nucleus, true nucleus, multinucleate condition, this point is important, is seen in plasmodium. Flagella is absent. Reproduction is maybe asexual or even it may be sexual. Now, I'll talk about the life cycle here. Now look here, I think the small diagram can help you children. I don't know how many of you can see it clearly. Hope that you see it clearly. I wish that you see it clearly. Now listen what I'm discussing here. These are the two amoeba-like cells. Think like amoeba-like cells. Plasmogamy occur, means protoplasm fuse. Karyogamy also occur, nucleus fuse, resulting in the formation of a diploid zygote. What happens to this? It undergoes many mitotic divisions. Means free nuclear divisions. In most of them, free nuclear means this diploid nucleus undergo many free nuclear divisions and develop into many nuclei. Then you're going to see a plasmodium. What is this plasmodium? It is a single cell surrounded by plasma membrane with many nuclei. This is plasmodium. This is the vegetative mass of slime mound. If you ask me, how is slime mound look like? This is how it look like. What are the ploidy? Diploid. Why? Because there are many diploid nuclei, because it is developing from zygote. So whenever conditions are favorable, whenever there is plenty of decayed organic matter, it starts spreading on it. All right, secreting slime. Hence it is called slime mold. Starts spreading on it. And this plasmodium spreads on dead and decayed organic matter. What? Conditions will not be same at all times, isn't it? All right. So therefore, when there is deprivation, when there is a lack of proper food, you know what this plasmodium does? It is going to produce, look here, a fruiting body or an erect mass called sporangium, just like fungi. All the diploid nuclei migrate into this sporangium. They undergo reduction division and give rise to spores. An interesting and most important point about the spores. They have a wall. Plasmodium does not have a wall. Mixed amoeba does not have a wall. Sporangium does not have a wall, but spores has. And that too cellulosic wall. That is a plant character seen in slime molds is a very popular question. Spores are windborne. They're highly resistant. They get liberated. And the spore wall break. 50% of them will develop into mixed amoeba of plus strain. The remaining 50% will develop into mixed amoeba of minus strain. This is very simple. You already know all this. Your tutor already discussed this all. All right, this is how it looked like, the life cycle in slime molds. So why you call it as plant, animal, fungal protist? Why you call a plant protist? Because it contains cellulosic wall. Why you call animal protist? Because there is no wall around plasmodium. And why you call fungal protist? Because just like fungi, there are sporangia and inside that spores. That is the reason why you call it as plant, animal, fungal protist. Slime molds, very important. Physarum is an example for all this is not needed, children. Sometimes you know what happens. These diploid cells, they form an aggregate. They don't fuse. They don't undergo free nuclear division. All these diploid cells may aggregate and form pseudoplasmodium. That questions, I don't think at this level it will be asked. All right. So if you look at the exam which was held on 3rd of May, one thing is very clear. They did not ask any questions outside your NCERT purview. Even the families like Asteraceae, Malvaceae, even they are not touched, right? So I'm not presuming that the same thing will happen on the 21st of June, but the probability of a similar type of question paper on 21st of June is more. So, but at the same time, I want you to read those additional points as well, children, to be on the safe side, because you have time now. All right. So therefore, plant characters, animal characters, I told you, pseudopodia, absence of cell wall around plasmodium, fungal characters, fruiting bodies, etc. Then I talk about protozoans, animal protist. Look here, I'm a plant associated with plants. I don't want to discuss much about protozoans, animal protist. Let your zoology master talk more about it. But because it is there in our biological classification, we'll be just touching and going away from it. All right. Common name, animal protist, habitat, aquatic and terrestrial. Cell wall is absent in all members. Chloroplast is absent in all members. Nutrition is only heterotrophic. Nucleus is generally one, but paramecium, 2 nuclei. This was asked in NEET exam about 6 years back. Flagella, there is only one in Trypanosoma and Leishmania. Reproduction may be asexual or sexual. What are the special features? They are divided into 4 types based on locomotory organs. They may be free-living or parasitic, flagella, cilia, and pseudopodia. Linnaeus kept them in animals, Whittaker kept them on Protista. Look at amoeboid protozoans, children. Well, just I don't want to talk more about it, just go through it. Nature is free-living or parasitic. Silica in marine forms. Then we talk about flagellated protozoans. I don't want to talk about it, just go through it. Free-living or parasitic, just like amoeboid. Outermost layer is pellicle. They have a single flagellum. Trypanosoma causing sleeping sickness and the vector is CC fly. Leishmania cause kala azar, the vector is sandfly. Ciliated protozoans. Now look at ciliated protozoans. Many cilia, they show coordinated movements. They may be terrestrial, marine or freshwater. Asexual reproduction not needed. Outermost layer pellicle. Opening is gullet, 2 nuclei per cell. Example is paramecium. Somewhere we come across paramecium even in molecular basis. Genetic code is nearly universal. All right, but in mitochondrial genes and paramecium, some of the nonsense codons code for some amino acids. All right, you know that. All right, some of the nonsense codons code for glutamine in paramecium. So that is the reason genetic code is no doubt universal, but there are exceptions, mitochondrial genes and paramecium. All right, now coming to sporozoans. Locomotion absent, habitat endoparasite, nature parasitic only, malaria causing. Come on children, this is, Oh, again Protista in brief. Come on, I'm little tired, so please go through this. We already discussed all this. Holophytic nutrition, holozoic. Anyway, not needed. That's it. Very diversified kingdom, Protista, we have completed. Took a little time for us, but I think it is worth taking because this is a kingdom where, there are some areas which you have to be very good at. And we found some ways to remember this very important kingdom called Protista. Now we talk about the most important kingdom. And in fact, by far the last one in this biological classification, all right, because what is there after that is viruses, hardly takes less than 2 to 3 minutes. But this is a kingdom where we are going to spend at least 10 minutes and try to understand this properly. So habitat, warm and humid regions. Linnaeus, the place, we'll start with the simple statements first, okay? Placed in kingdom Protista. Whittaker classification, placed in kingdom Fungi. Vegetative body is mycelium, and the mycelium is made up of hyphae. Those branches are called hyphae. And how are the hyphae? This is very important. Aseptate and multinucleate in Phycomycetes. In Rhizopus, Muca, Albigo, the entire mycelium doesn't have any septa, and that is the reason why they are multinucleate. Very, very important. Remember this. Septate, uninucleate, and binucleate in Ascomycetes and Basidiomycetes. I'll tell you why it is like this. Septate and only uninucleate cytoplasm is seen in Deuteromycetes. So that's about the fungal body, children. Okay? Habitat and fungal body. Now we'll talk about the cell structure in fungi. All the points are in fact known to you. Cell wall is made up of chitin and polysaccharides. Cellulose is also seen in some of the Phycomycetes members. All right, so polysaccharides are also seen along with chitin. All right, but chitin should be given more importance. Plasma membrane, lipids and proteins. Cytoplasm is a semi-fluid. Plastids are absent. Ribosome, 80S. Of course, in mitochondria, it could be 70S. Reserve food could be glycogen or oil. Nutrition, mostly saprophytic, parasitic or symbiotic. And very important are these 2 examples of symbiotic. Mycorrhiza, it's a symbiotic association of pinus and fungi. Corollariza is seen in cycas and blue-green alga. That's also symbiotic. This is mycorrhiza. I'm sorry. These are the 2 examples which you have to remember. Coralloid roots, cycas and blue-green alga. Mycorrhizal roots, pinus and fungi. All right. Lichens are also example for symbiotic. Alga and fungi association is seen in lichens. Fungi and pinus association is mycorrhiza. Remember this. All right. Enzymes are secreted and the food is digested outside the mycelium, extracellular. This is the living reaction occurring outside the body. Yesterday we were discussing about that. Living reactions can occur inside the body and outside the body. And outside the body, example for that is extracellular digestion of fungi. All right, and the digested food is ingested by diffusion. Vegetative reproduction is by fragmentation, children, in all 4 classes. Fission and budding is given in your textbook, but they did not tell you in which organism you're going to see this, only in yeast. And this is the very important part of the syllabus, children. In Phycomycetes, asexual spores are sporangiospores and zoospores. In Ascomycetes, it is conidiospores. In Basidiomycetes, asexual spores are absent. Whereas in Deuteromycetes, asexual spores are conidiospores. Okay? Now the most important, sexual reproduction. It could be plasmogamy, followed by karyogamy, followed by meiosis. Plasmogamy ante protoplasm fusing. Karyogamy means nuclei fusing, followed by reduction division. So these are the 3 steps or the 3 stages of sexual reproduction in fungi. Even this is important. Criteria for classifying fungi into 4 classes. Morphology of mycelium, mode of spore formation, and fruiting bodies. Remember this, very popular question. All right. Now, if we come back to sexual reproduction, children, anyway, when I go to Phycomycetes, I'll discuss this in detail, but just for a minute. Plasmogamy, karyogamy occur immediately. Okay, when the 2 gametes fuse, protoplasm fuse, nucleus fuse. That results in the formation of zygospore, diploid. Other name for zygospore, oospore, diploid. Remember this word. And reduction division occur in it, resulting in the formation of germ spores. And these spores develop into a mycelium. Okay? Very easy way. I told you isogamy, anisogamy, and oogamy. All 3 are seen in Phycomycetes. Gametes are formed. The gametes fuse, protoplasm fuse, nuclei fuse. Results in the formation of zygospore or oospore. Reduction division occur in them, resulting in the development of spores. Then we talk about Ascomycetes and Basidiomycetes. Here also same. Plasmogamy occur, karyogamy occur. But first plasmogamy occur. But the nuclei are reluctant to fuse. They want to stay separate. All right. Then that cell with separated nuclei will divide and redivide and develop into a mycelium, which is dikaryotic. After producing fruiting bodies, inside a special sac-like structure called ascus in Ascomycetes, now the 2 nuclei fuse, karyogamy, immediately for the reduction division, producing ascospores. Same way in Basidiomycetes, after a prolonged time gap, karyogamy occur in a club-like structure called basidium, followed by reduction division. So point to understand is, in Phycomycetes, Ascomycetes, and Basidiomycetes, plasmogamy, karyogamy, and meiosis, all 3 occur. But in Phycomycetes, plasmogamy and karyogamy occur in immediate time intervals, immediately one after the other, resulting in the formation of diploid zygospore or oospore, followed by reduction division. Whereas in Ascomycetes and Basidiomycetes, long time gap exists between plasmogamy and karyogamy, resulting in a dikaryotic phase. That point is important. Very good assertion, reason, question. Dikaryotic phase is seen in Ascomycetes and Basidiomycetes. Reason for that is a long time gap exists between plasmogamy and karyogamy. And what are and Deuteromycetes, you know, children, there is no sexual reproduction. That is the reason why they are called fungi imperfecti. And coming to the sexual spores, the same thing. Ascomycetes, ascospores, Basidiomycetes, basidiospores, Deuteromycetes absent, Phycomycetes, germ spores. I think we already discussed the introduction part very nicely. In the sense, we brought some important aspects, all right, into proper perspective. So first we talked about the fungal body, how it looks, mycelium hyphae, and how the hyphae looks. Phycomycetes, it is aseptate, that point is important. And in Ascomycetes and Basidiomycetes, it is uninucleate and septate and binucleate and septate. Then I talked about the cell structure. The most important one will be the cell wall chitin. It contains N-acetyl glucosamine. All right. Then we talked about asexual reproduction, vegetative reproduction, and sexual. Vegetative, fragmentation and binary fission and fragmentation that is seen in binary fission and budding that is seen in yeast, And then we talk about asexual spores, very important. Sporangiospores and zoospores are seen in Phycomycetes. Conidiospores are seen in Ascomycetes and Deuteromycetes. And asexual spores are generally absent in Basidiomycetes. Coming to sexual spores, germ spores, zoospores, germ spores, zygospores, or oospores are seen in Phycomycetes. Germ spores are haploid, zygospore and oospores are diploid. Ascospores are sexual spores produced inside the ascus in Ascomycetes. Basidiospores are sexual spores produced outside the basidium in Basidiomycetes, and sexual spores are totally absent in Deuteromycetes. Nice facelift we gave to this chapter. It is just a formality for us to get rid of the 4 kingdoms, 4 classes now. We'll go a little faster and get rid of this chapter on high. Coming to Phycomycetes, common name algal fungi. Along with chitin, they also contain cellulose and falls. Habitat, there may be some members like saprolignia or aquatic. There are some aquatic members also in this. They may grow on decaying wood, damp places, and they may be obligate parasite. This is very important, albigo. Albigo cause white rust in crucifers. That's a very important point to remember. Mycelium may be aseptate and sinusitic. Very important point to remember. Vegetative reproduction is by fragmentation. Asexual reproduction, already we talked about. Sporangiospores are inside the sporangium, non-motile. And zoospores are motile. In slime mounds we studied, no? Sporangium, all the diploid nuclei migrate, reduction, division. But here there is no diploid nuclei. All the haploid nuclei migrate and form a wall. And that is called sporangiospores. And if they have flagella, you call them as zoospores. Both of them are produced inside sporangium. Remember, both of them are endospores. All right, both are endospores. Sexual reproduction, true sexual reproduction producing gametangia and zygospore. Look here, I have shown here. These are the gamete producing. Gametes fuse to form zygote, 2N, also called oospore. It undergo meiosis and produce spores, and these are germ spores. They get liberated. So plasmogamy and karyogamy occur in immediate succession, resulting in the formation of zygospore. Zygospore undergo reduction, division, and give rise to germ spores. Very easy to remember. Example, Rhizopus bread mold, muca is similar to Rhizopus but lack rhizoids. And look here, which diagram is there in your textbook? It is muca, not Rhizopus, in your textbook diagram of NCERT. Remember that. Albigo, very popular question. White rust. Don't get confused between rust, wheat rust and white rust. Wheat rust pathogen is Basidiomycetes. All right. So that is Puccinia, wheat rust. All right. Whereas white rust pathogen is albigo, which is a Phycomycetes member. Late blight in potato, which caused Irish famine, all right, is Phytophthora. The people started concentrating on plant diseases for the first time when the entire Ireland went into famine because of this pathogen, which devastated the entire potato crop. All right. So anyway, that's a different story. It's not in the NCERT book, not that much important. Special characters, I told you, aquatic, sinusitic hyphae, this is important. Endogenous asexual spores. Plasmogamy and karyogamy occur in quick succession. Isogamy, anisogamy, and oogamy. Diploid zygospore or oospore. Zygotic meiosis. Meiosis occur in zygote. Sexual spores. That's nice. We have completed Phycomycetes in the best possible way. Now we go to Ascomycetes. Common name sac fungi because the ascus look like a sac. Habitat coprophilous. They may grow on dung. That's the reason why it's called coprophilous. Parasitic or saprophytic. Mycelium unicellular, multicellular, septate, and monokaryotic, and later dikaryotic. I'll explain this. Vegetative reproduction is by fragmentation. Asexual reproduction is by conidiospores. They're exogenous. Sexual reproduction, a primitive type of sexual reproduction producing sex organs, namely antheridia and ascagonia. Do I have a diagram here? Yes. Now look here, children. Look at this diagram here. These are the spores. What are the spores? Ascospores. Ascospores of plus and ascospores of minus strain. They develop into a primary mycelium. Look at the primary mycelium. Primary mycelium is monokaryotic. They are septa. It's not clearly visible, the septa, but they are septate. And it develops into a male sex organ behind antheridium. So I am ascospore. I develop into a mycelium, septate, uninucleate. And at the tip of it, I produce a single cell called antheridium. I am minus strain ascospore. minor strain ascospore. I also develop into a primary mycelium, septate and monokaryotic. And the terminal cell of me becomes ascogonium. Up to here, no problem. Now look what happens. The nucleus from antheridium migrate into ascogonium after protoplasm fuse, means plasmogamy is occurring. But the nucleus from antheridium migrate into the ascogonium, but will not fuse with the nucleus of ascogonium, means it remains dikaryotic. This dikaryotic cell, look here, this dikaryotic cell will later develop into many celled cup-like body that is said to be ascocarp. Can I call this as mushroom? Yes. Morels and truffles are costly mushrooms of Ascomycetes. It means that the ascocarp primarily is made up of dikaryotic mycelium. And the dikaryotic mycelium is forming a cup-like body, and that is said to be a mushroom. What happens later? Some cells, especially at the top of this mushroom, is long and sac-like. Now in those cells, the two nuclei now fuse. Actually, the nuclei should fuse here in between antheridium and ascogonium. Only protoplasm is fusing, but not nuclei. Dikaryotic cell is formed, no fusion. Dikaryotic mycelium is formed, no fusion among nuclei. Cup-like mushroom is formed, no fusion among the two nuclei. After they produce a sac-like ascus, now the two nuclei fuse, resulting in the formation of a diploid nucleus. That undergo reduction division and produce 4 ascospores, which later become 8 inside the ascus, endospores. And those spores get liberated. 50% of them will develop into ascospore of plus, and the remaining 50% ascospores of minus. What plus strain ascospore produce? Primary mycelium with antheridia. What does minus strain ascospore? Primary mycelium with ascogonia. Protoplasm fuse but not nuclei. Dikaryotic cell that will undergo many mitotic divisions and form a cup-like ascocarp. The terminal cell of ascocarp or mushroom becomes ascus, sac-like. The two nuclei now fuse, karyogamy. Reduction division is followed immediately after karyogamy, resulting in the formation of ascospores. Very nice. In fact, you know all this. Your teacher would have explained this all right in detail. Saccharomyces. Examples are important, children. Examples in Ascomycetes are important. Remember this. It's a unicellular fungi. Then Penicillium. It's an antibiotic producer. Ripening of Roquefort cheese also we use Penicillium roqueforti to increase the taste and flavor. All right, so we are going to sprinkle the Penicillium spores, all right, such that they germinate and secrete some chemicals which increase the taste and flavor. And Roquefort cheese is very popular in Europe, especially in France. Neurospora is an experimental organism. One gene, one enzyme hypothesis was also worked on this, Beadle and Totem. Aspergillus, citric acid producer. In microbes chapter it is given. Claviceps, it causes ergot disease in rye. Morcella, morels, they're costly mushrooms. Tuber, truffles, they're also costly mushrooms. Gibberella, Bacchanet disease. I brought some of the examples of fungi from other chapters into it. All right, Monascus, statins, they're cholesterol lowering agent. Once you understand Ascomycetes, Basidiomycetes looks like a cakewalk because it is exactly similar to Ascomycetes. I'm talking with regard to sexual reproduction, except that there is no antheridium and ascogonium. Directly, the vegetative hyphae fuse, and again it forms a dikaryotic mycelium. That will not become a fruiting body. That produce aerial fruiting bodies called mushroom, stalk and a cap, and you're going to see a club-like basidium, the two nuclei fuse. That's a very simple one. We'll be discussing that very fast. Common name, club fungi. Basidium is club-like. Bracket fungi is polyporous. Puffballs is lycoperidon. Remember, bracket fungi and polyporous also belong. Soil, logs, tree stumps. These two are examples very important. Puccinia and Eustilago. Eustilago cause smut. Sooty charcoal-like appearance on plant parts is smut. And Eustilago. Puccinia, rusty symptoms, dark brown, rusty marks on leaves is wheat rust causing, and that is Puccinia. Remember these two. Eustilago cause smut, Puccinia cause rust. White rust means albigo. That's a Phycomycetes member. All right. Mycelium may be septate and monokaryotic. We already discussed ascospore monokaryotic primary mycelium. Same here also. Basidiospore, monokaryotic primary mycelium of plus strain, monokaryotic primary mycelium of minus strain. Later, the protoplasm fused but not nuclei, resulting in a dikaryotic mycelium. And later, you are going to see the development of fruiting bodies. Clamp connections and dolypores are seen in dikaryotic secondary mycelium. Outside chance for this question to come. Vegetative reproduction by fragmentation, asexual reproduction is absent. Look at it, what I've said. Primitive type of sexual reproduction without sex organs, gametangia and zygospore. It is called somatogenous copulation. Plus and minus strain hyphae undergo plasmogamy but not karyogamy, resulting in haploid dikaryotic mycelium. It later produces fruiting bodies called basidocarp mushrooms, and fruiting bodies are absent in smut and rust fungus. That question cannot come. In a mushroom, on the lower side of the cap-like structure, you see gills, and the gills possess a club-like structure called basidia. Karyogamy and meiosis occur in quick succession within basidia. It results in the production of exogenous sexual spores on sterigmata, and these are called basidiospores. Look here. Did I overdid by talking this again and again? I get that feeling. But anyway, plus and minus strain monokaryotic mycelium, plasmogamy occur but not karyogamy. Dikaryotic mycelium. This dikaryotic cup-like fruiting body called basidiocarp. This dikaryotic club-like cell called basidium will be there in that fruiting body. Karyogamy and meiosis occur inside the basidium. Exogenous basidiospores, they germinate to give rise to plus and minus strain. This is it. You could see that. Examples: agaricus, white button mushroom. Amanita, it's a toadstool, it's a poisonous mushroom, amanita. All right. Eustylago, smut, puccinia, rust, lycoperidon, puffball, polyporous bracket fungi. We got rid of ascomycetes and basidomycetes. If at all I ask students which part of biological classification is a little difficult, they always say fungi. But I don't think this is difficult anymore. Already you know everything about it. All right. That now we have properly revised this topic. So therefore, there will be no problem at all in answering questions. Biological classification, children, of later in NEET, the number of questions are less. But if you go back to 2018, 2019 and 2020, or even 21, there are at least 5 or 6 questions coming from this chapter. So be careful about that. All right. So see that you remember all the examples properly, and see that you remember the different types of spores, especially in fungi, and those points are important. All right. Coming to Deuteromycetes. Common name, fungi imperfecti. Habitat, dead and decayed organic matter. Mycelium may be septate and branched. Vegetative reproduction is by fragmentation. Asexual reproduction is by conidiospores. There are no sexual stages, children. But if some Deuteromycetes member show a sexual stage, then you know what we do. We are not going to keep under Deuteromycetes. Why? Because we have committed ourselves by saying that it is fungi imperfecti, means no sexual stage. If any members of it produce antheridia, ascagonia, plasmogamy occur but not karyogamy, then you see a dikaryotic fruiting body called mushroom, then you see a sac-like structure on the top, the 2 nuclei fusing. If it is similar to ascomycetes, then what you do, we are going to transfer this into ascomycetes with a separate name. I'm going to give an example. As long as a fungi called fusarium is reproducing only by conidiospores without any sexual reproduction, it is kept under Deuteromycetes. When the same organism started producing fruiting bodies showing sexual stages similar to ascomycetes, then this is shifted to ascomycetes with a separate name called Gibberella fusicori. So it's very clear that when any Deuteromycetes member show any sexual stage, then it is shifted either to ascomycetes or basidomycetes, because Deuteromycetes members does not show any sexual reproduction. That's enough. More than this is not needed. Early blight in brinjal, colitotrichum, red rot in sugarcane, trichoderma. In microbes chapter you studied about cyclosporin A, immunosuppressive agent or biocontrol agent. Oh, come on. This looks a little boring for you now because you have already gone through all this. All right, so just run through this. Coprophilus, major decomposers, Deuteromycetes, tree logs and stumps, Basidomycetes. Aseptate mycelium, phycomycetes, very important. Septate mycelium, all except phycomycetes. Monokaryotic, all except phycomycetes. Dikaryotic, very important. Ascomycetes and basidiomycetes. Clamp connections, only basidium. Fragmentation, all four. Zoospores, phycomycetes. Sporangiospores, phycomycetes. Conidiospores, this point is important. True sexual reproduction, phycomycetes. Sexual germ spores, phycomycetes. Zygospore, this point is important, phycomycetes. Isogamy and isogamy oogamy, phycomycetes. Gametangel copulation, phycomycetes. Zygotic meiosis. The word zygote is used only for phycomycetes. That could be the reason why I have written only phycomycetes. Plasmogamy, karyogamy, and meiosis, all except deuteromycetes. Long time back, this is very popular. Plasmogamy and karyogamy, ascomycetes and basidiomycetes. Dikaryotic stage, only these two classes. Fruiting bodies called mushrooms, ascomycetes and basidiomycetes. Mycosymbiont and lichens, ascomycetes. Did I make a mistake? Mycosymbiont in lichens, I'm sorry, it should be ascomycetes and basidiomycetes, right? I made a mistake here. All right. So endogenous motile asexual spores, phycomycetes. Exogenous asexual spores, ascomycetes. Endogenous sexual spores, ascomycetes. Exogenous sexual spores, basidiomycetes. Sex organs are seen in phycomycetes and ascomycetes. No sex organs, but sexual reproduction is seen, basidiomycetes. Neither sex organs nor sexual reproduction, deuteromycetes. Sometimes when we dissect things a little deeper, you get a feeling that this topic is difficult, but actually it is not. All right, you understood everything when your teacher was discussing this. And today, not only we discussed it one more time, we have also already done some worksheet with regard to every kingdom. That I think is a very good thing we did today. All right, isn't it, children? Yes. Now we going to wind up now in less than 3 minutes. Just hold on. Viruses, the last point. All right, with regard to viruses. It is not included any kingdom by Whittaker. It is ultramicroscopic, acellular, obligate parasite. There are no cells, obligate parasites. Means they always parasite. Look here, virus is not feeding on you for water and food. No. Virus is feeding on you for its multiplication because you have ribosomes. Viruses doesn't have any cell, no ribosome. It cannot produce protein. It is feeding on you because you have some enzymes which can help its genetic material multiplication. So when I say viruses are parasites, not for food and water, it is for their multiplication. They are depending on protein synthesizing machinery of your cell. Enzymes for replication of their genetic material. Understand that. Ivanovsky called viruses as filterable agents. After B comes C. Bijonic called it as contagium vivum fluidum. After B comes C, he coined the term virus. Remember that? He took a diseased leaf, rubbed on healthy leaf. The healthy leaf also suffered because the diseased leaf contained virus and that got spread. You know, all viral diseases, not all, majority of viral diseases are highly contagious. You know pretty well about that. Stanley, crystallization of TMV. These are the non-living characters, children. No cells, absence of metabolism, not influenced by antibiotic, cannot be cultured in artificial medium outside the cell, absence of intrinsic motility. What are the living characters? Presence of genetic material, ability to multiply in living host, mutations can be seen in its genetic material, disease-causing ability. Then nucleoprotein, protein coat capsid, it's made up of capsomeres and it may show helical symmetry, spiral arrangement or polyhedral symmetry. All right, example for polyhedral symmetry is adenovirus, respiratory ailments it causes. In microbes chapter we studied. And in bacteriophage, the head shows polyhedral symmetry and the tail shows helical symmetry, the two types of symmetries. It's a complex virus. Genetic material in a virus could be either DNA or RNA, not both. QB bacteriophage, TMV, poliovirus, flu virus, corona, AIDS, and many plant viruses, single-stranded RNA. This is 5. No, I just sometimes, I can't really type properly. So 5x174 bacteriophage is single-stranded DNA. Lambda phage and animal viruses, double-stranded DNA. Wound tumor virus and rice bark virus is double-stranded RNA. This question is not needed to remember. Bacteriophage lambda, tadpole-shaped, hexagonal head. It contains double-stranded DNA. Plant diseases, mosaic, weed clearing, leaf roll, leaf curl, chlorosis. Animal diseases caused by viruses, smallpox, chickenpox, polio, measles, flu, hepatitis, AIDS, corona, mumps, and herpes. What are the uses of viruses? It can be used as vector in genetic engineering, all right, and a biocontrol agent, baculoviruses, all right, they eliminate specific arthropods and insects. Prions, scientist name is not needed, Prusna, highly folded protein. They're small, they're similar to the size of viruses. Mad cow or BSA in cattle, and an analog disease in humans, Creutzfeldt-Jakob disease. Coming to viroids, not include any kingdom, just like this scientist name, remember, Diner. Small single-stranded RNA, it has less molecular weight. So viroids are smaller than viruses. Prions are similar to viruses. Potato spindle tuber disease and citrus exocortosis disease are caused by prions. All right. Then lichens, not included in any kingdom by Whitaker. Nature, cells, but did not find any place in Whitaker's classification. Plant board is a colony of algae and fungi. Algal members could be Chlorophaceae or Cyanophaceae. The fungal members could be Ascomycetes and Basidiomycetes. Do they fix atmospheric nitrogen? Yes. Are they pollution indicators? Yes, of sulfur dioxide. Are they pioneer community? Yes, on rocks along with mosses. Examples, Cladonia is a reindeer mass, Roxella, litmus producing lichen. Took a little time, but we successfully completed biological classification without leaving a single letter of your NCERT textbook. And that is all for today, children, with biological classification. Tomorrow I'm going to come again with a different chapter, not difficult, different chapter called Plant Kingdom, and we'll make sure that we remember this chapter, Plant Kingdom, in best possible manner, just like as we did for biological classification. Tata for today, and we'll meet again tomorrow. Bye and thank you.